everybody, myself Ramesh. Especially today we will talk about very brief introduction about the food microbiology. So especially in food microbiology, we will discuss briefly about the foodborne pathogens, meat spoilage and microorganism. And we will see the slide by slide. You can see before we are going to discuss about the food microbiology is one of the branch of especially in food chemistry, or you can say this is one of the study about the microorganism to inhibit and create or the contamination of food. Food microbiology, especially it is the one of the general biology. It is discussed about the microorganism found in the food, including their growth and characterization, identification and pathogenesis. Especially we will think about the meat. So meat especially spoiled by the microorganism because of it contains a lot of nutrients, growth factors and its pH, and meat of range is 5.6 to 7.4. Especially you can see the meat range of pH. This is very important. Why? Because some competitive exams they will ask in various competitive exams and even the medical science also. So what is the pH of the meat is 5.6 to 7.4 is the range of pH, and microorganism for the soil, water and manure make of the dominant flora of meat. And during the slaughterhouse, the external surface of the animal may contaminate the meat by the direct contact or through the above source of the equipment and personal and slaughterhouse area. Now you can see the next slide. Before we are going to discuss about the detail about the food microbiology, the two terminal points or the two terms are very important. What is the aerobic condition and another one is the what is anaerobic condition. What is aerobic condition means presence of oxygen. You can see any reactions or any changes, something is happen in the presence of oxygen, that is called aerobic condition. And what is the anaerobic condition means without oxygen. So that reaction will goes or that changes will happen, that's you will call as the anaerobic condition. Aerobic means oxygen presence, anaerobic means without oxygen. These points please keep in your mind. So now we can discuss about the major source of meat contamination. So how the meat will contaminate. So the first is the thickness of stick, knife and animal hide and the gastrointestinal tracts and the hands or handlers and contaminants, handling and the storage of environmental lymph nodes. These are the few important major source for the meat contamination. Now you can see here why the meat change in color. So general question, meat is the major source of protein is the myoglobin. So the major part is the myoglobin. So because of it contain the red color. Why the meat was contain red color? Because of the myoglobin protein are the responsible to majority of the red color. So purplish color when it is mixed with the oxygen because of oxyhemoglobin and is produce the bright red color. So the meat from the golden animals will be darker in color because of the myoglobin level increases with the age. So next we will discuss about the how the myoglobin changing to oxymyoglobin in presence of different factors. We will discuss in future slides. And now you can see cause of meat discolor. What are the major problems or what are the major cause to change the meat has discoloring of the meat. So however the exposure of the storage light as well as the continued contact of the myoglobin oxyhemoglobin or oxymyoglobin with oxygen leads to formation of metmyoglobin, the pigment that turns into brownish red. This color change does not means the produce of spoilage. So myoglobin is the heme iron containing protein. So myoglobin was the internal part, one of the major part, the center part is the iron heme containing protein. So it gives this color and it is different of the dietary of iron. So myoglobin stores oxygen in a muscles and cell and similar hemomyoglobin are stored in oxygen blood cells. So these are the hemoglobin contain meat contains and darkness will be appears in the color. So hemoglobin and myoglobin both are the storage of oxygen in blood cells, and these are the major proteins in the meat sample. Now we can see the next slide. The myoglobin color changes depending on what meat interior temperature also is one of the factor. And when it is the meat cooked when the proteins denature and recombine with coagulant or the meat becomes a opaque or whitish color. So you can see that this is the raw meat brown, it's changing the near end of the shelf life. So these are the factors affecting of the meat. It has a discoloring of colors or white stuffness on the meat. You can see observe in the market, few of that meat market, you can see white stuff you can observe in the meat surface. So because of slimy lines of the cause because of mold and growth of surface of spoilage sticks. So meat and meat products are important source of human infections with the variety of foodborne pathogens. So microorganisms generally found in meat. What are the microorganisms? Bacilla, Salmonella, Staphylococcus, Mycobacterium, tuberculosis, Plasmodium. These are the major microorganisms you can generally find in the especially in meat. One is the Bacilla, Salmonella, Streptococcus, Mycobacterium and tuberculosis and Clostridium. These are the microorganisms you will find in the meat. Next you can see here the spoilage of microorganism Archobacter, Pseudomonas bacillus and the Staphylococcus and Aspergillus and Mucor penicillium. These are the molds. These are the spoilage of meats and molds are the example of for the meat spoilage. So raw meat start deteriorating by the own enzyme and microorganism chemically oxidize that fat. So these are the factors you call as that how meat spoilage. Now you can see the what are the factors determining the spoilage of meat. So intrinsic factor, extrinsic factor. So you can see factors are mainly two types to the finding of the spoilage of meat. One is the intrinsic factor, another one extrinsic factor. So intrinsic one is the pH, water activity, redox potential, antimicrobial substance. And what is the extrinsic factors? Temperature, relative humidity and atmosphere. These are the major two factors, intrinsic and extrinsic factors to affecting of the meat spoilage. Now you can see here the industrial production of microorganisms that make them. There are the enzyme, microbial cell, antibiotics, steroids, alkaloids, food additives, cosmetic and chemicals. They are expensive. They are using nowadays citric acid. Many they are using in microorganisms. And the poultry meat and the control of meat-borne pathogens. These are the few I was cutted from Google, and you can see some papers and the meat-borne diseases also you can find. You can read it, go through it. You can find out what are the bacteria, especially Campylobacteria, Salmonella. These are the major you can find in the meat spoilage. And you can see here the processed meat, how the process will meat, what are the lactic acid in bacteria will role. These we will discuss in next class in detail. Now you can see spoilage under the aerobic condition. I told you what is aerobic condition means, that is the presence of oxygen. What are the spoilage? And these are the some gram negative bacteria and gram positive bacteria and yeast and non-filamentous fungus. You can see the actobacteria and the many bacteria, bacillus and campylobacteria. These are the few bacteria or you can microorganisms involved to fermentation process. And you can see that environment, agent and host. These are the main factor, the triangle factor to affecting on the meat spoilage. This is the foodborne disease and dynamic interaction, agent, hosting and environment. These are the triangle factor, especially the meat spoilage. Next you can see microbial spoilage of meat and meat product. You can see how that meat sources, hen, ducks are different and what are the microorganism image. And you can see that the treatment plant A and plant B, some microorganism and microbiology studies, carcass, and you can see primals and the retail stakes market, and you can find out the microbiology parameters like water activity, color, sensory analysis. These are that. And you can see major cause of food spoilage, physical, chemical, microorganism and others. You can see especially physical factors of food spoilage, temperature, RH and light, mechanical damage. And you can see especially chemical, enzymatic reaction, non-enzymatic reaction, rancidity and chemical interaction. The bacteria, yeast and molds are the major microorganisms. Bacteria, yeast and molds are the major to the food spoilage, and the other are the insect, rodents and the animals and birds. And you can see here, you can see other what are the new nitrates, nitrites and composite regulation, crude meat products and quality safety regulation you need to follow, processing, packaging, many you can see here. And you can see actually the image 1 and image 2 how that meat was adding. You can see that here how the oxidation and reduction was going on. You can see that here this is the one meat and this is the adding of oxygen, it converting, removal of oxygen, it changing like. So that is called as the process. The meat always will be, you find out by the oxidation and reduction. So myoglobin and nitric oxide, nitroso myoglobin bright red and nitroso hemochromic the pink color. These are the, you can the flow chart how the meat was happening in the changing of its color, the oxidation and reduction, especially in myoglobin, nitric oxide, it to nitroso myoglobin, nitroso hemochromic pink color. And you can see the composition of meat bones and meat pigments. Myoglobin is and the lesser content of hemoglobin. So the greater concentration of myoglobin is the raw meat and more intense in the bright and red color because of the myoglobin is the major part, lesser part is the hemoglobin. And you can see that how these oxyhemoglobin, deoxyhemoglobin, myoglobin and metmyoglobin. You can see these are the structures and how these, you can see the color oxymyoglobin, deoxymyoglobin, metmyoglobin metals. And the oxygen level and the meat color, you can see high oxygen levels, so that is called oxymyoglobin, and these are low oxygen levels, that is called metmyoglobin, deoxymyoglobin. These are the oxygen levels in meat color. And you can see especially this is the one of the paper I was taken this picture. I like this picture. The structure changes in the myoglobin. You can see especially this is the control of myoglobin amplification and the peripheral ring, these rings and goes under the deoxyhemoglobin, and then removal of oxygen, it changes the oxymyoglobin. Then after you go under the oxidation, then it change in the methionmyoglobin, methane myoglobin, or simply you can see oxidation reduction process. This is the actual the structure of myoglobin changes in the, especially in meat product. And you can see the factor affecting of meat color, chemical states of myoglobin, the ferrous Fe+2, water interact purple, deoxymyoglobin, oxygen in red color, oxymyoglobin, and nitrate, the unstable pink color, nitric oxide myoglobin, and carbon dioxide, carbon monoxide, CO, that red color, carboxymyoglobin. These are the ferrous reaction. This is the ferric, means Fe+3, water, and the brown color. And these you can see that how this H2O2 hydrogen peroxide, SS sulfur hydrides, sulfomyoglobin, these are the factors affecting of the ferrous and ferric changes. And you can see actually the protein responsible for the meat color of myoglobin. This is the one of the nice picture. You can see globin free binding sites. These are the deoxymyoglobin and these are the oxymyoglobin status. And you can see these are the drying methods, how that coloring and the description parts you can see clearly. And these are the different factors how the protein will the meat spoilage and changes. And you can see this is how the raw meat and drying and the biochemical reaction, dehydration, proteolysis, lipolysis, formation of metmyoglobin, nitrosomyoglobin, enzymatic activity, dried meat, how this biochemical reaction, then after you can see the dried meat. The food spoilage is the major part, and food spoilage are the nowadays big problem for each and everyone peoples. And you can see how the microbial growth in especially in food product, and how the protein, polypeptide, peptides and amino acids, how the protein change in according to the microorganism. And chemical changes caused by the microorganism you can see clearly, and the organic acid others. And this is the testing activity spectrum of, you can see how this is the strict antibiotic producer across to the side chains. How suppose if you are going to that microbial studies like the antibacterial studies, and the antibiotic diffuse into the agar, and Staphylococcus is the cell mass and cross sticks in the test organism. Then you can incubate and you can find out what the, this is the actual procedure for antimicrobial. If you like this, like, share, comment and subscribe my YouTube channel Thank you for your great attention to listen my class. The next class we will discuss about in detail about the how food